All right, good morning, everybody, and thank you for joining us for our first webinar of 2021, the Salmonella and Listeria species in-depth webinar. We're going to go ahead and get started here with a quick introduction. My name is Lauren Ellie. I am the technical liaison here at Cherney Micro, and along with me is Nicholas Hesse, and he's going to be answering any of your questions, so be sure to utilize that question and chat function throughout the webinar. Interaction is definitely encouraged, so please ask questions, offer comments. Like I said, this session will be recorded for others to view later. So we'll jump right into Salmonella here. Salmonella, as some of you might know, is part of the EB family. EB testing is typically used as an indicator organism for the potential presence of Salmonella. So if you look at that picture on the slide on the top left there, you'll see the EB family in that circle, and circled there in red is the Salmonella species. So Salmonella bacteria is also a rod-shaped bacteria. We call that bacillus. The pictures on the bottom left and bottom right are depictions of what bacillus rod-shaped bacteria look like. Salmonella also has flagella, which you can see in those pictures, which means it is a motile bacteria. It is also a gram-negative bacteria. For those of you who aren't aware, gram-negative bacteria, as depicted on the picture on the right, is used with gram staining, which is a very effective and helpful tool for those of us in the lab. And it's able to tell us a little bit more about what type of bacteria we're dealing with. So gram staining is going to tell you a little bit more about the structure of the cell wall of the bacteria. So in that picture, you can see the difference in the cell wall between the gram-negative bacteria and the gram-positive bacteria, where the gram-negative just has that one thin line of peptidoglycan, that one thin line of purple, versus the gram-positive, which has a much thicker layer of peptidoglycan in that cell wall. So when we do gram staining in the lab, gram-negative bacteria are going to stain as pink bacteria when we look at them under the microscope, and gram-positive bacteria are going to stain as purple when we look at them under the microscope. So Salmonella is structured, and this cell wall is going to be gram-negative, so it's going to look like the left side of that photo there. Salmonella is also a facultative anaerobe. And just for clarification, if you're not familiar, when we talk about bacteria, we talk about whether it's an aerobic bacteria or an anaerobic bacteria. So anaerobic bacteria means that it is able to grow without the presence of oxygen, whereas an aerobic bacteria is able to grow with the presence of oxygen. So Salmonella being a facultative anaerobe means that it can grow with both the presence of oxygen and without the presence of oxygen. The Salmonella bacteria is divided into two different species, and although it just has two species, it's got thousands of different serovars, which we'll get into here in a minute. So those two species that I had mentioned, Salmonella enterica and Salmonella bongeri. So Salmonella bongeri is usually linked to illnesses in cold-blooded animals, most frequently associated with reptiles and rarely found with human infection. So it's not one that we discuss all too often. We're much more concerned with Salmonella enterica. So Salmonella enterica has 6 different subspecies listed up there. And you'll notice those Roman numerals as we go 1, 2, 3A, 3B, 4, and 6. You might wonder what happened to the 5th. Salmonella bongeri was previously classified under Salmonella enterica. However, it was different enough that it was classified as its own species division. So bouncing back up to that Salmonella enterica, it is the human pathogenic species, and it has over 2,600 different serotypes. So for those of you not familiar with serotypes, you might be wondering what the heck I'm talking about. So we're going to dive a little bit deeper into what a serotype is and how it helps us understand Salmonella a little bit better. So like I said, Salmonella enterica has over 2,600 serotypes. Some examples that you might be familiar with is Salmonella typhimurium, Salmonella Newport, Salmonella enteriditis, and typhimurium, Newport, and enteriditis are all referring to the serotypes. So what the heck is a serotype? A serotype is a term that's describing the cell surface of Salmonella. So more specifically, the antigens or molecular structures that are present on that cell surface. So when we're talking about serotypes, we're using the cell surface to describe different types of bacteria based off the antigens present on the cell surface. So typhimurium, Newport, and enteriditis all have different cell surface structures, and that's how we're able to tell them apart based on serotype testing, which is usually done with various anti-sera solutions as depicted in the picture in the bottom right. So those solutions will react with different types of Salmonella and be able to narrow it down and tell us what group of Salmonella we're talking about here. So it gets us a little bit more information to tell us if we're looking at Salmonella typhimurium, Newport, or any of those other species and serotypes of Salmonella. So I just wanted to briefly touch on that, just so you understand that even though there are only 2 different species divisions, 6 subspecies, we're actually talking about a very diverse group of bacteria, because when you consider all those different serotypes, there are a lot of different types of Salmonella, not just those 2 species. So we're going to talk a little bit about the Salmonella growth conditions. So they grow in a temperature range of 43 degrees Fahrenheit all the way up to 115 degrees Fahrenheit, although their optimum temperature for growth is 98.6 degrees Fahrenheit. And if that sounds familiar, it should, because that is the typical temperature of the human body. So Salmonella grows very well. It grows within pH ranges from 4.1 all the way up to 9, and its optimum pH is going to be right in that neutral range, about 6.5 to 7.5. The lowest water activity, AW stands for water activity, required for growth is 0.93. So I'm going to say that again. The lowest water activity for growth is 0.93, and 0.93 is considered fairly high. Salmonella likes to grow in the presence of moisture. However, Salmonella can survive in as low as 0.2 water activity. So that means that Salmonella is able to survive in those dry products, like if we're talking whey powder, infant formula powder, dry pet foods. Salmonella is definitely able to survive within those conditions. So we'll talk a little bit more about factors that are affecting Salmonella survival. They're definitely capable of adapting to extreme conditions. So they can survive that low water activity, various pHs and temperatures. So their tolerance to heat is enhanced with a neutral pH and a low water activity. So, and survival is also enhanced with colder temperatures and lower water activity. I'll direct your attention over to the graph on the right-hand side. So what's shown here is a dry sucrose product that's been inoculated with Salmonella. And so on the bottom of the graph, we're talking about storage time in weeks. And on the y-axis of the graph, we're talking about how much Salmonella is present. So the sugar was inoculated with levels of Salmonella, and you can see as we go further and further out, those top 2 lines up there, that Salmonella level stays fairly consistent. And those top 2 lines are when it's stored at refrigerated temperatures. So when stored at refrigerated temperatures, that Salmonella can definitely persist all the way out to 52 weeks. Those bottom 2 lines are stored at room temperature, where you can see the Salmonella dies off around 28 to 36 weeks. So the survival of Salmonella is enhanced with colder temperatures at low water activities. Salmonella is also adaptive to becoming antibiotic resistant, which can cause a lot of trouble. And Salmonella is known to persist in the environment and in product for years. So there is a study that cites that Salmonella can survive in honey for up to 28 months when stored at 50 degrees Fahrenheit. That's over 2 years that it can stay present in something like honey. That's quite a long time that I wouldn't necessarily anticipate bacteria being able to stay for that long. So it's kind of surprising how Salmonella can find ways to survive. So you're going to want to look for Salmonella in your environment. Salmonella bacteria are found in the GI tracts of many species, humans and animals alike. Salmonella can find its way into the environment via feces contamination. Salmonella can persist in water supplies and soil and on various surfaces. So all of these pictures here where you've got a green check mark, you have the potential for Salmonella. So in the top right corner, if you've got standing water on the roof of one of your facilities and you happen to have a roof leak, you could have possibly introduced Salmonella into your facility. Salmonella can grow in the soil. Salmonella can also be tracked into your environment, into your plant on the bottoms of shoes. There definitely can be present in drains, but just as a reminder, don't only think of the areas that look visibly dirty, because they can also find their way onto production floors and near your food contact surfaces. So it's very important to survey your entire environment, have a very strong environmental monitoring program, and be on the lookout for Salmonella. So Salmonella and product. Traditionally, Salmonella has been associated with animal products, but it has been linked to other products such as fresh produce as well. Recently, I've seen some Salmonella recalls for mushrooms, onions, fruit products. And then, as mentioned earlier, Salmonella can survive well in those low moisture foods, such as spices, peanut butters, other nut butters, pet foods, etc. Cross-contamination has occurred between raw and ready-to-eat foods. So it's very important within your plant that you control traffic flow, especially between your raw ingredient areas and your finished product areas. So I wanted to go a little bit into what we do when we receive customer products here at Cherney. So when we're doing Salmonella pathogen testing, if it's a product, we're going to aseptically weigh out your product for the amount that is needed. We'll add an enrichment broth specific to Salmonella growth, such as buffered peptone water. That'll incubate for about 24 hours at 35 to 37 degrees Celsius. So depending on your method and testing platform, that initial enrichment is either transferred to a secondary enrichment to help further grow that Salmonella, or it's ran directly on the platform that you chose. For Salmonella environmentals, it's similar. So we'll use those environmental swabs that you guys have sent in to us. We'll enrich it with broth, incubate it for 28 hours, and then it's ran directly from that sponge with the broth on the requested platform. So if we've done that for you and you received a presumptive, got a call from us, and now you're thinking, well, now what? What do I do? So on the left side, if it's an environmental presumptive, you're gonna obviously want to clean the area. Nobody wants to keep Salmonella around in their plant. But you can use that information to track and trend those presumptives throughout your environment. After initial positive, it's a good idea to consider doing vectoring samples. So pictured on the left there, the red would be your initial site where you got that first hit for a Salmonella presumptive. And you would want to consider branching out in all directions to see if you've got contamination in other areas nearby. So it's not only looking directly to the left, to the right, you really want to consider like a 360 approach and get a good picture of where else you might be seeing some Salmonella contamination in those areas. However, on the right-hand side, if you've got a product presumptive, your next steps are largely going to depend on the procedures and practices that you have put into place at your specific plant. However, for positive product, we do perform a confirmation, and many times customers will request an additional biochemical identification just to verify that that Salmonella is really there present in that product. And I have pictured there a chromogenic agar with Salmonella growing on it. It typically looks that strong purple color. Just thought that was kind of fun to include in here so you guys can see kind of what the lab sees when we look for Salmonella. So then I wanted to talk briefly about Salmonella illness. We've got greater than 1.2 million cases annually in the US, more than 23,000 hospitalizations and 450 deaths. So I have this map of the US up here, and I just wanted to talk about how widespread just one outbreak can be. This was from a live poultry outbreak from back in 2017 that lasted all the way from January to August. And it definitely just doesn't affect one region. It doesn't affect just a couple people. It has the capability to affect the entire nation. So I just want to stress, how important our jobs are, how important your jobs are. It really can't be understated how important food safety is, and the effect that the things that you guys do, the effect that we have, that can really affect the entire nation. So it is very important. And I just wanted to stress the importance of food safety here and how detrimental it can be to some people. Talking a little bit more about the specific illnesses that we see. There are two types of illnesses, typhoidal and non-typhoidal salmonella. So this typhoidal one, we've got about 2,000 cases annually. It's a very serious sickness. These symptoms include a high fever, diarrhea, constipation, aches, headaches, and lethargy. So the two types of salmonella that cause this are Salmonella typhi and Salmonella paratyphi A. And these serotypes are only found in humans. And this type of illness is typically associated with drinking contaminated water that could be contaminated with untreated sewage, where you get that salmonella bacteria in the water. And the infective dose for this type of illness is fewer than 1,000 cells. It's very serious, mortality up to 10% if left untreated, and you have a prolonged onset period. So 1 to 3 weeks up to 2 months from your initial exposure, and you could be feeling this very serious illness. So if we talk about non-typhoidal, this is what you mostly associate with your food poisoning and not feeling so well. So there's over 1 million cases of this annually. It's gastrointestinal. You're going to be getting some nausea, vomiting, diarrhea, cramps, fever, and these are caused by the other serotypes, not Salmonella typhi and not Salmonella paratyphi A. It lasts a couple of days, usually over in a week. Healthy individuals don't require treatment. However, it can be troublesome in those who are immunocompromised. So the infective dose is as low as 1 cell. It doesn't take very much to give you food poisoning and make you feel like crap. So the rapid onset, it can be as soon as 6 hours from exposure all the way up to 72 hours. And I wanted to discuss a little bit about Salmonella recalls. So Salmonella contamination can happen anywhere from farm to table. So it can happen in your production facility, manufacturing, distribution and delivery, and at home with your preparation and consumption of foods. So talking about farm, in 2010, there was chicken and feed contamination that resulted in a recall of 500 million eggs. In 2011, with manufacturer, we had some tainted turkey burgers that recalled 35 million pounds of ground turkey following illness in 10 different states. Could even be in your delivery and distribution system. In 1994, there was contaminated ice cream when raw unpasteurized eggs were hauled in 1 or more tanker trucks, and then later that carried the pasteurized ice cream premix. And so obviously those two things don't mix and happen to sicken over 200,000 nationwide. And it can definitely happen to you at home with preparation. 2007, frozen pot pies sickened 35 people due to undercooked product. And as a manufacturer, you want to make sure that you're providing the consumer with appropriate instructions on how to cook your product. And obviously at home, we need to follow those instructions so that we make sure that we have safe food. So that wraps up the Salmonella portion. So we're going to transition now into the Listeria species. So going back to those basic Listeria characteristics, is also a rod-shaped bacteria like Salmonella was. It is bacillus. It is non-spore forming, And unlike Salmonella, it is gram-positive. So I put that same picture up and you can see that the gram-positive is that thicker peptidoglycan layer in the cellular structure, which means it would gram stain under the microscope as purple. And that helps us again in the lab to kind of identify what type of bacteria that we're dealing with. The Listeria genus is very fast growing. More and more species are still being discovered. So Listeria genus first came on the scene as a foodborne pathogen in 1981. From 1981 to 1992, only 10 species of Listeria were known. During 2020, there were 17 species total that had been identified. So the picture below kind of shows the different types of Listeria. People are probably more familiar with that group on the top, highlighted in green. So the main Listeria members are L. mono, Listeria coelegii, Ivanovii, Welshmeri, Inocua, and then marthi. And Listeria marthi was first described in 2010. So that one's one of the new members. So you'll notice that in that green box over in the picture, there are two that are written in purple. So Listeria mono, as well as Listeria Ivanovii. Not a lot of people know this, but Listeria Ivanovii is also pathogenic. However, it only affects and causes illness in ruminants. So we're talking like cattle, sheep, and it is not considered pathogenic for humans. So, however, on the other hand, Listeria mono is a very large concern for us. It is pathogenic in humans. So it is our primary pathogen focus when we're talking about Listeria food safety. L. mono has 13 different serotypes. And remember, when we're talking serotypes, I'm talking about the cell wall structure. And the serotypes that are most often affected with foodborne illness are 1/2 A and 4B. So Listeria growth conditions, the temperature range is 33 degrees up to 122 degrees Fahrenheit. The optimal temperature is going to be right around 86 degrees Fahrenheit. pH range 4.5 to 9.6, and the lowest water activity for growth is 0.92. So Listeria definitely likes moist conditions. Unlike most bacteria, Listeria is salt tolerant and able to grow up to 10% salt and is able to survive in 20 to 30% salt. Also, unlike most bacteria, Listeria actually grows in cold environments. I'm not just talking like where I was with Salmonella, where there's enhanced survivability. Listeria actually grows in cold environments. So think coolers, ice and condensation pans and lines, anywhere where it's wet, Listeria has the capability to grow. So Listeria present in your environment. Listeria can enter the plant by raw materials, by movement of people and different equipment. The Listeria genus is resistant to environmental conditions like high acid and salt, cold temperatures like we just discussed, low humidity and oxygen. So Listeria is able to persist in your environment for multiple reasons. And one of those main reasons being biofilm. If you're not familiar with biofilms, it's when a collection of bacteria is able to grow together and produce polymeric substance that's like a slimy residue that protects those bacteria, and they're able to grow and proliferate, and then eventually they're able to continuously reintroduce themselves into the environment through dispersion after that biofilm has matured. So that slimy sticky substance, the polymeric extracellular matrix that I mentioned, helps those bacteria become more resistant to sanitizers. That slimy substance acts as a barrier that most sanitizers are not very efficient at penetrating. And biofilms, you'll find those in certain niches, definitely in your cracks, nooks, crannies, anything like that. Listeria and different biofilms are more likely to form in untouched areas where you don't routinely clean. So those hard to reach areas, that's where you'll find a lot of biofilms and different Listeria growth. So jumping back in, dealing with persistence here, you're going to want to make sure you have a hygienic design of your plant and your equipment. You'll have effective cleaning and sanitation, like I said, an effective environmental monitoring program cannot be understated. It is extremely important. You want to make sure that your personnel are practicing good hygienic practices. And then you'll want to control the traffic patterns of people, materials, and equipment throughout your plant. So if we talk a little bit about Listeria and product, high-risk foods are going to have those intrinsic characteristics that support the growth of El Mono, neutral pH, high water activity. Some of those frequent contaminations are listed on the right there in that picture: sprouts, deli meats, hot dogs, smoked seafood, soft cheeses, unpasteurized milk. Contamination can occur in that wide range of food types, definitely not limited to just one type of item. So frequent outbreaks have resulted from contamination via the environment, either during your manufacturing process, during your processing, or during the packaging of that product. Sporadic cases and outbreaks of listeriosis have generally been associated with those ready-to-eat foods that are extendedly held for periods of time at refrigerated temperatures, which is going to allow, like I said, Listeria is able to grow in those cold temperatures. So breaking it down here at the lab level, what happens when you send us your Listeria testing? So if we've got some Listeria product for you, we're going to weigh out aseptically the appropriate amount of product, enrich it in a broth specific to Listeria, usually demi-Fraser or UVM broth. And after that initial incubation, it's transferred to another broth specific for Listeria growth, Fraser broth. And then it's incubated for an additional 24 hours at 30 degrees Celsius. For your environmental enrichments, very much the same, where we're going to take that sponge sample, enrich it in a Listeria broth. It's going to incubate for 40 to 48 hours and then ran on your testing platform. However, there have been some PCR methods with a proprietary media that's going to allow for a little bit shorter incubation when ran via PCR testing. So if you've got a presumptive, you got a call from us and we notified you. It's kind of that same thing. If you found it in your environment, you're going to want to track and trend. And then it's always a good idea to do that vectoring. If it's a presumptive product, it's going to depend on what procedures you have in place. We'll automatically start a confirmation and make sure that Listeria is there. And many times customers will also request to do a speciation because they're concerned, is it El Mono in my product? And in there in that picture is from an Aloha auger plate where Listeria will grow as a blue color and the El Mono will have that halo around it. So it's easy to differentiate between the different species of Listeria genus on that type of auger plate. Talking about Listeriosis outbreaks, there are two different types of illnesses, invasive and non-invasive. The infection in invasive develops and spreads to your nervous system. Symptoms may include the headache, stiff neck, confusion, loss of balance, and convulsions. A little bit less serious would be that non-invasive gastrointestinal kind of mild symptoms with a fever, muscle ache, nausea, vomiting, what you typically think of when you think of food poisoning. However, it is very serious if pregnant women ingest Listeria monocontaminated food. And the picture on the right-hand side, if a pregnant woman were to ingest El Mono contaminated food, it would get absorbed through the intestine into the bloodstream and then could potentially affect through the placenta the fetus, causing miscarriage. So it's extremely important for pregnant women as well as immunocompromised. This could have very serious repercussions if they were to ingest Listeria monocontaminated food. So the infectious dose for these is unknown. It is suspected that it's less than 1,000 cells in susceptible people. Estimated lethal dose for humans is 1.9 times 10 to the 6 CFU. That's just under 2 million cells. So a little bit about Listeria Recalls. In 1985, we had some Listeria contaminated queso fresco, sickens 142 people, killed 10 newborns, 18 adults, and caused 20 miscarriages. In 2011, we had some Listeria contaminated whole cantaloupes, sickened 147 people across 28 different states, caused 33 deaths. 2013, we had some contaminated pre-cut celery in chicken salad that was served at hospitals and sickened 10 people who already had serious health problems, and 5 of them died as a result. 2015, there was a popular ice cream that was contaminated with Listeria, caused 10 hospitalizations and 3 deaths. 2020, we've had some contaminated deli meats, hospitalized 12 people across 4 states and caused 1 death. And we do have a recent outbreak going in 2021 for El Mono in soft cheeses, queso fresco cheeses, among several others. So it does continue to be a problem for many different food types. And that concludes our webinar for Salmonella and Listeria in depth. Thank you guys so much for joining us. Please make sure you tune in for our other scheduled webinars. We have pathogen detection platforms where I'm going to go in depth on how we detect those pathogens. That's scheduled for April 21st at 10 a.m. We've got serial dilutions and results on June 2nd. And we're going to be understanding a little bit more about nutritional analysis on August 11th. And all of those webinars are at 10 a.m. And if you've missed any of our previous 2020 webinars, they are still available for viewing through our website. The different topics are listed below here, and also check out the blog section of our website for some more fun and educational discussion topics. If you wanted more information specifically about biofilms, there was one released yesterday, so you can go to the website there and check it out and read up on it. And I will be taking questions. If you guys have anything about Salmonella and Listeria, just let me know.